Good morning, everyone. My name is Claudia. I am a PhD candidate from the Department of Biochemistry in the Medical School of Ribeirão Preto at the University of São Paulo. I would like to invite you to welcome our first presenter for today's webinar. Okay, so Marco Brocca is a biologist. He has worked for 20 years for Tecniplast, developing an entire aquatic product line. He has visited many aquatic facilities. He has talked to many principal investigators, to many facility managers and technicians in order to develop innovative solutions for multiple aquatic species. Okay, so I would like to welcome you, Marco. So feel free to start your presentation.
Muito obrigado, Claudia. Thanks a lot. Thanks to everyone. Just let me know when you see the presentation. Can you see? Hello?
Not yet. Now it is okay for me.
Okay, can I start the presentation?
Yes.
Hello? Can I start the presentation? Can you see it?
Yes, you can.
Okay.
Yes.
Thank you, Claudia. Thanks for the nice introduction. As you said, I am Marco Brocca. I work for Tecniplast. Tecniplast is an Italian company that is based in the north of Italy. Here is exactly where we are, in the Pre-Alps and in the lake region, so in the north of the country. And this one is an aerial view of the company itself. As you can see, we are a manufacturing company. This one gives us the unique opportunity to develop products internally, from the idea to the final product, which is something very, very interesting. For example, here you see the production of one of the mold that we do. So from the idea of a tank, we start developing the drawings and then the mold itself, which will give life to a real tank. As you can see, here you see an injection molding machine that, using the anthropomorphic robot and very complicated molds, we can reach a very high level of product and also surface and finishing of the product. Once the product is made, we can also do all the final quality checks of all the products. For example, here you see one sensitive needle that is touching edges of one of our tank, just checking if the shape of the tank corresponds to the real and the final product we would like to obtain. Tecniplast is a fairly large organization with over 600 employees all over the world. In many different regions, we have dedicated company, and where we don't have dedicated companies, we work with distributors. For example, in Brazil, our local partner is called STEC. But out of this large group of employees, Tecniplast has a group of people that are fully dedicated to aquatics. And we are about a team of 20 people, which ranges from design engineers, project manager, product managers, sales and technical managers, which are just, all those people are just dedicated to the development and the support of the aquatics product line. In going into the different products we develop, I would like to take you into following a specific project that we have completed in Southeast Asia a while ago. This one is a fairly large facility that shows a quarantine room with approximately 150 tanks, a main room which has two separate aquarium rooms, one here on the north side and one here on the west side. The one on the north side has approximately 2,500 tanks. We do also 3D layouting, as you can see here, when we do large installation, but also for small ones. We always now try to work with 3D programs that allows all the users to have a real idea of the space. Here you see the potential expansion on the southwest side. Here there is a zoom in of the installation. Here you see the area where the centralized life support is positioned, this one here, while here you have the main aquarium room with 2,500 tanks. The life support space was pretty small, so we had to fit in the proper life support that is able to provide the best water quality with the right amount to all the tanks. Imagine that in less than 10 square meters, exactly in 8.8 square meters, with a height that is lower than 2.5 meter, we have been able to design and install a centralized life support that is able to provide over 60,000 liters per hour to all the fish tanks. So very small space, high efficient life support. Here you have a 3D view of the life support. Those are the programs that we use. For example, here you see the self-cleaning drum filter. Whenever we can, we tend to use self-cleaning drum filtration that avoids consumables. Here you see stainless steel UV lamps, specific biotowers. Here you see an extraction of the unit. So we canceled the main sump, and you can see all the internal parts. These one are submersible pumps, a set of 6 pumps with 2 pumping line with 3 pumps each. Here you see 316L stainless steel or titanium meters, vertical pumps, and we can check with all those programs that all the components are fitting well together. Once we have designed and built the unit, what we do internally is also the wet test. So all the times we do provide a unit like this one, we assemble it in our facilities, and we do a wet test of the unit. We also develop dedicated software, as you can see here. This is the one of the unit you just saw, where we have immediately very visible the main water quality parameters, pH, conductivity, temperature, the water level, how much water you're changing, and all the main water quality parameters that are necessary to check the well-being and the water quality inside your system. Moving on, for now, in many installations, people are requiring an automatic feeding system. This is what we call Tritone. Tritone is a robot that is able to distribute the proper diet to the proper amount of fish automatically using this specific dispenser, and you have up to 4 different dry diet dispensers, so you can distribute 4 different types of dry diet, plus a liquid diet that sits here at the bottom of the robot, and the robot is able to, reading specific barcode label, to distribute the proper amount of diet to the proper age of the fish, depending on your fish population. We have installed nowadays over 220 units all over the world. Those labels are electrostatic, so you can reuse the labels multiple times, and Tritone can also be equipped with a dedicated camera that allows you to check from your PC portable device how the Tritone is feeding your animals. All the tanks that has a label are then fed with the proper amount of diet according to the age of the fish. Here you see an installation that we did a few years back in South Africa, and with this unit we have now a very high performance mechanical action of this unit. You are able to remove all the dirt, algae, and biofilm very successfully from your tanks. The first one we've developed was called 650A. We have installed over 35 units all over the world, but everybody was asking for something cheaper and something smaller. So we have launched Calypso, that is a dedicated aquatic washer, very small in footprint. This was launched in 2017, but very, very efficient. Here you see how the tanks are positioned inside the machine on dedicated trays. You can have large tanks, medium-sized tanks, small tanks. And the unit, using a sliding door, allows you to load the tanks very easily. At the bottom of the machine, you have the container with all the necessary chemicals. So far, we have installed and are in use over 50 washing machines all over the world. Here, there is a quick movie that shows you how this machine works. As we said, is the only validated aquatic cabinet washer in the world. When we say validated, means that the unit has been proved to remove very effectively and very safely the dirt from the tanks, also the biofilm, without the need to pre-scrub the tank. So no possibility to damage the tanks during the washing. Here you see how the water and chemicals are covering all the parts of the tanks without leaving any spot not covered. After the washing cycle, we make sure that all the chemicals are removed through a very efficient cycle using reverse osmosis water. Not only you can wash tanks, but you can also wash all the other parts in your aquatic facility, nets, lids, baffles, and this one is done very efficiently. Not only we saw feeding, washing, tanks, filtration, but we also focus on breeding because we know it's one of the most important aspects in a zebrafish facility. So we have developed different products for breeding, very small breeding tanks, the 0.7 liter one, medium one, 1 liter, a very large system to collect lots of embryos called ice spawn. But more recently, we have developed two other products, a smaller ice spawn called ice spawn S, 13 liters, and a medium breeding tank, which is called sloped breeding tank that has a volume of 1.7 liter. Here you see the number of fish that you usually, or people are usually putting in those containers. We can range from 2 to 200 fish, collecting from few hundreds up to thousands of embryos. We will just focus now on the slope breeding tanks that has been baptized by someone Sex on the Beach breeding tank, and we will see why. Here you see a side view of the unit. This breeding tank has an internal shape that is very specific, has a sloping internal part with a long beach area where the fish can go on the shallow water and perform their natural breeding behavior. In fact, if you see from this picture, all the embryos are collected where the shallow water is. Here you see you have also a divider to keep males and female separated. And here you see the fish enjoying the shallow water with all the eggs collected at the bottom of the tank. If you look carefully here, you see that the bottom of this internal tank is opaque and is gravel-like. So they have a very specific texture that the fish can feel. Here you have a quick movie that shows the behavior, the breeding behavior of the fish, all in the shallow water when they're ready to breed. And now you see the fish that, thanks to the mating courtship, are able now to release fertilized embryos just on the shallow part of the tank. This tank is now pretty famous and widely used all over the world. We have done facility of different sizes all over the world. Here you see a gigantic facility that we built in USA where you have two main areas for Rex, and here you have the parts where the CLSs are, all on the same floor. Here you see another one in Japan, fairly large with the automatic feeding systems. Here you see another one in Germany where all the tanks are one floor above the filtration that sits underneath. In this case, we have two systems here. And this one is a picture that shows you the sizes of the life support, providing the best water quality to the fish tanks. This one is another interesting one that we did in UK, which is the opposite. All the tanks and Rex are sitting one floor below the life support, and life support is sitting above here. In this case, we had to use a two pumping systems. But not only we do very large installation, we also do very small installation. In this case, this one is what we call standalone. So one single rack system where the filtration sits underneath and you have a dedicated monitoring for the operator to manage the water quality and the entire fish colony just using your fingers. Here you see a section of the filtration system that sits underneath here, where we use a drum filter in this case as well. So this one is the only product available that is consumable free. So no need of pre-filter or mechanical filter to handle this unit. Here you see a better image of the touchscreen where all the main water quality parameters are always provided, pH, temperature. You can add a dissolved gases probe in case you would like, and conductivity. These systems allow you also to download all the data, for example, alarms or water quality parameters, and you can also access the main touchscreen from remote using a portable device or your phone. In this case, this one is an interesting installation that we did in South America, in Bogota, in Colombia, in which this one is pretty interesting because this unit sits at a very high level above sea level, over 2,500 meters, and so far we had no issues with the animals swimming in those tanks. So we are able to maintain the proper oxygen level even at these altitudes. Another specific system that we developed is the heat shock standalone. So very similar configuration from the one that you saw before, where the filtration sits underneath here. But in this case, you are able to increase the temperature, the water temperature, up to 40 degrees and start activating heat shock proteins. Here you see how it works. Usually you keep the temperature at 28. Whenever needed, in this case, the set point was at 37. You have a ramp-up phase, an exposure phase that you can decide how much time you want to keep this temperature, and then a cool-down phase, which is the phase C. And this one is done just using your finger and controlling this unit. Here you see a standard heater, and here you see the enlarged and more powerful heaters inside this unit. All the components and the software has been developed in-house. Very easy to set, as we said, just using your finger, you can set the proper temperature level. Another interesting one that we developed is called TIME, or Toxicology Integrated Modular Element. What is this one? This one is a standard standalone, as you can see here, with an extra small rack, in this case with 5 rows, that you can use with 3.5 or 8 liter tanks, which can be used as a flow-through module. So you can add to your standalone a dedicated single module that can be used as a flow-through system. This one can be used as a quarantine option if you want to keep your quarantine fish on a flow-through base, or as a toxicology experimental added module, because the water that goes into this tank goes in and goes out, doesn't go back into the main recirculation of water that goes with the standalone. Or if you're running toxicology studies, we also have the so-called toxicology rack, a fully flow-through system that has 3 rows. Each row is separate from the other. Using dedicated solenoid valve, you can decide if you want to push water in 1 row, 2 rows, or 3 rows independently. You cannot control the flow. The flow is controlled automatically, but the water, again, is not recirculating, goes in and goes out, goes to the waste. So if you want to do repeated toxicology studies in 3 different levels, using 3 different types of chemicals, you can do in specific systems. As you can see, and you will see later on, we try to develop in-house, following your specifications, different type of products. If you have a very small facility, you are just starting using Zebrafish, you would like to validate the model, and you don't have, or you have a limited budget, we have recently developed the so-called ZEBTEC Super Mini, a very small, compact unit, 3 levels, different tanks, small, large, very small, able to, anyway, house over 500 fish, so a good number of fish can be used for small labs, startups, labs, or quarantine as a quarantine system. It is very simple, but it has everything that a good system needs to have. Temperature control, automatic water exchange. The UV lamp is 316L. All the filtration needed sits underneath. So small budget, very powerful system. Of course, we have different type of tanks, as we said, 3.5 with divider, larger 8 liter. We use the patented self-cleaning technology. So each tank has a sloping front wall, sloping bottom, lower point of the tank here. The siphon is active in helping removing all the debris and maintaining the best water quality in your tanks. We have developed very small tank, 1.1 liter. With the same concept, we have developed the 2.4 liter tanks. So if you think to stick together two 1.1 liter, you reach a 2.4. This one is a unique tank that can be split into 4 different areas if you want to isolate single fish. On the same level, you can have 1.1 and 2.4 sitting one after the other. Our unique nozzle that with the on-off mechanism and the regulation that is separate from the on-off. We have developed genotyping solution with drawer style trays that you can remove, and you can isolate single fish where the tanks are sitting in a water bath. So maintaining the proper temperature, but not investing the recently clipped fish with a very high flow of water. So we can develop multiple and different solutions for your needs. Okay, all those tanks are very nice for all the experimental protocol. But what's next? What do we know about zebrafish? We know that zebrafish is a fish that like to be in groups. They like to be in groups and move in schooling. So showing this shoaling behavior of the fish. So what we have developed is a dedicated tank called Z-Park, the best housing, according to us, for housing large colonies of adult zebrafish. What is Z-Park? Z-Park is a large tank, 16 liter of volume, where you can house in one single tank over 150 fish, up to 200 fish. And we have built a dedicated mold to mold this tank in the same thermoplastic of all our tanks. Here you see the tank sitting on the rack where you see the behavior of the fish. Look how the fish are moving. The water flow comes from the bottom, not from the top. You see the behavior of this large group of fish that are swimming against the current, and they are dragged on the top of these long and elongated tanks. Very, very interesting to see the behavior. So it's an enriched tank. It is great for adults. What else can this tank do? Okay. So if you thought of the large tank that we had, it has a length of 25 centimeters. The Z-Park, with 16 liter of volume, has over almost 3 times the length of a standard tank. Why this one is important? It's important because this allows the fish to swim longer distance horizontally. Very important for zebrafish. But not only is it important to house them in the best possible manner, also to keep the colony healthy, you need to collect embryos from them. So we have developed a dedicated in-tank spawning platform that allows you to collect embryos whenever you want. Here you see the behavior of those fish sitting in this tank. When you have a large population of fish, maybe not all of them are ready to spawn the eggs at the same time. So if you slide the spawning platform, just the fish that are ready to spawn the eggs can go in the shallow area of the tank and show the natural spawning behavior on this dedicated platform, as you can see here. So naturally, you can collect the embryos, keeping a very healthy colony of zebrafish, and always excellent water quality during the spawning because the water flow always is guaranteed. Now we have the best colony of fish. We have spawned them, but we also need to collect the embryos. So if you want to collect the embryos efficiently, there is a sliding embryo tray that allows you, without disturbing the fish, to collect the embryos very efficiently. The fish are not net, are always healthy, stay always in the tank with the best water quality. We also have a divider if you want to keep males and females separate, or if you want to keep the spawning platform separate from the fish. And when you're ready, you remove the divider and the fish are exposed to the spawning platform. We have also learned that zebrafish prefers to have a dedicated background at the bottom of the tank that is gravel-like, and this one has been demonstrated through a dedicated test that has been done, published paper, showing that zebrafish prefers to have a background that is rocky, gravel-like. So we have developed at the bottom of the tank dedicated runners on top of which the tanks are sitting, providing the best environmental enrichment for the animals. In many countries, you also have the need to disinfect the wastewater. So what we did, we have developed dedicated disinfection water that are using different methods. We can disinfect the water before being released to the ambient, to the waste, using chemicals, using UV, using heat, so warming the water up, or a combination of all of them. Here you see some example of those units. We can do them of different sizes. This one, for example, is a very large disinfection unit able to warm the water up to 90 degrees, so a pasteurization, and then it is cooled down and released to the waste. More recently, we have also developed Interzeptec. What is Interzeptec? Is an environmental biological detector. So if you want to do health monitoring, there was no standardization. Someone was using fish and sending sentinel fish. Someone was using sludge. Someone was using debris, biofilm, but no standardization. So we wanted to standardize the health monitoring of your colony. So what we have developed is a dedicated tool that is called Interzeptec that allows you to collect what? Debris and biofilm in a very efficient manner from your system. And you can choose which is your biological unit. It can be a set of tanks, can be a rack, can be a system. How does it work? Interzeptec. Interzeptec is made of a column of different cylinders or caps, at the bottom of which there are specific filters. On the top one, there is a very large mesh that is used as a pre-filter. At the lower stages, there are two cones with very fine mesh sizes filters, those one, the two in the middle. At the bottom, there is an empty one that is used to collect biofilm on the walls of these cylinders. This column of filters is made of a specific plastic material that we have studied that is able to increment the formation of biofilm. So we want to be sure that also biofilm is in the Interzeptec. Interzeptec columns of caps comes in a dedicated container. The container is installed on the return water from your racks, for example, in this case. The water goes into the different filters and goes back into the sump. Approximately, an Interzeptec needs to stay a minimum of 5 weeks, normally 5 to 8 weeks. It's filtering up to 450 liters of water per day and is collecting detritus and sludge. After that, what you do, you simply open the container, you put your filtering tower in a Falcon tube, and you send to your diagnostic lab to identify what is in your system. Each Interzeptec comes in boxes of 5 each, singularly packed and irradiated in order to be sure that they are sterile. We did many tests. For example, this one is one that we did a while ago of the length of 1 year, checking the Interzeptec every 5 to 7 weeks, and the Interzeptec was able to collect lots of pathogens that were in this facility, for example. Different type of mycobacterium, Pseudocapillaria tomentosa, and also Pseudoloma neurophilia. But we do systems, as we said at the beginning, not only for zebrafish. In this case, we develop a very large facility for axolotl, so a totally aquatic amphibian. In this case, an urodele. So you see the layout of the facility. This one is the filtration. This one is dedicated software that developed for this unit. And we developed systems that were able to accept all the stages of axolotl, from early larvae that are grown in these specific boxes, up to medium-sized muets that were housed in the dedicated genotyping tray, singularly housed, with the bottom perforated tank, as you can see here. And this one is the installation for the muet. Why this perforated bottom is important? Because they are very delicate, especially the breeding organism. And when you feed them, all the feces and debris are not staying in the tanks, but are falling through those holes and are collecting at the outside of the tank. So within the tank, we have the best water quality. While the animals are growing, we are putting them in larger tanks, as we saw. Here you see some of the 8-liter tanks with fairly large adult axolotl. But also we have dedicated, and here you see all the batteries of tanks and racks, but also we have dedicated, using larger tanks, spawning tanks for axolotl, with which we were able to collect all the embryos, using grass as well as an enrichment, embryos from the axolotl. So we were able to go from embryos to embryos, taking care of all the different housing capabilities and needs of the axolotl. And here you see the very large very large animals in a large plastic tank. Another very interesting project that we did on another different species was with octopus. So we developed for this facility two different types of standalones, or two standalone systems here. So here we switch from fresh water to salt water, so we can do saltwater system. This one is the design we did with all the filtration sitting underneath here, with 4 tanks at the top of this filtration. But not only, this system can be enlarged up to 8 tanks. So if in the future they want to rise the system, adding extra 4 tanks, you can do it. This one is the 3D drawing of the 8-liter tank, of the 8 tanks. Here you see the tank that we developed. Very interesting, all fiberglass with a clear window in the front to check the health status of the animals, clear lid that you can open just half of the lid. The water enters from here and goes out from the back here. As you can see, in order to avoid that the octopus could cover all the drain of the tanks, we have designed and dedicated separators with multiple holes, at least more than 8, otherwise with the tentacles they can close the holes, in order to be sure that the drain is always free. These are the drawings of the tanks, and these one are the tanks that were made, as we said, in fiberglass with clear lid that you can open with a locking mechanism, because octopus are very smart, with a frontal clear window as well. This one is another interesting one, just to give you an overview of the different type of system that we are doing. This one is a system for aquatic snails. So there is a tray here with a totally recirculating system. The snails are housed in this tray, but whenever you want, or the researcher in this case was able to put this cover on top of it that you can open and check the snails in case they would like to, because they're doing studies on different photo periods for the snails. Another interesting application that, according to your needs, we have been able to develop. More recently, we are doing not only plastic tanks, not only fiberglass tanks, but also glass tanks. This one is a fairly large installation that we did recently for cichlids. And as you can see here, you see the animals housed in large glass tanks. And the customer was so happy because cichlids are quite difficult to breed, but in this case, they've been breeding a lot. You see here inside the tanks, all the small cichlids, fish, and small fries that are swimming in the tanks with the adults. Last but not least, we do also a course. We just finished the 9th annual International Zebrafish Husbandry course that was from the 27th till the 30th of September. Is limited in number, is a group of people, 35 attendees, that are grouping up from different countries in the world, and for 3 days and a half, we just talked and learned the latest techniques on zebrafish husbandry. This is important if we want to rise the level of husbandry of zebrafish. Here you see a picture of the attendees last week, and we have people, as we said, from 13 different countries in the world, from Australia to Canada. A very nice and interesting course. Just to finish, we have done just to give you some ideas or some numbers, over 800 installations all over the world that are still active in over 50 countries all over the world. As we said, from South America, Brazil, wherever, North America, Asia, Europe, Africa, and Australia, New Zealand. Those are some of the species that are housed in our systems, from vertebrates, invertebrates, Daphnia, stickleback, guppies, lobsters, teleost fish, xenopus, amphibians. We are able to do systems for saltwater, freshwater, of any type, of any size. And with this, I would like to thank you a lot for this information. Here is my email address in case you would like to ask any question. If you don't have time now, feel free to ask them whenever you want. And just to remind you that there will be now a pool for having 3 of our breeding tanks given free of charge to you if you have taken part to this pool. Once again, thank you very much. Molto obrigado per l'atenção. Thanks a lot.
Thank you so much for the very nice presentation, Marco. Okay. I would like to ask if you guys have any questions, so Marco can answer now. I don't think so.
As I said, I left there my email address,
so
they can write me whenever.
Yes. All right. Thank you very much. So, guys, before we go to our next presentation, I would like to change the language now for Portuguese, okay? Because since you know that Marco works for Tecniplast, the company is providing a contest, yes. So I would like to change the language to Portuguese because it is going to work only for Brazilian people, okay? Então, pessoal, aqui a gente vai fazer o nosso sorteio de aquários de reprodução de Zebrafish, da Tecniplast, ok? Só para lembrar que o pessoal que for participar do sorteio tem que estar ao vivo para poder ganhar o prêmio. Ives, você vai compartilhar já para fazer o sorteio já? Só um minutinho.
Bom dia, pessoal, tudo bem? Eu vou compartilhar aqui. Cadê a Priscila? Priscila, tá por aí?
Bom dia.
Priscila.
Tchau, Marco.
Bom dia a todos. A gente vai fazer o sorteio.
Great presentation. Congrats.
A gente vai fazer o sorteio rapidinho aqui. Já está tudo preparado. Vou compartilhar a minha tela.
Vamos lá. Vamos ver quem está com sorte no dia de hoje.
Até agora tivemos 195 comentários. Vamos conferir os comentários.
Queria até aproveitar já, Ives, se você me der uma licencinha, só para agradecer em nome da STEC e da Tecniplast pela participação de todos no nosso sorteio. A gente ficou muito, muito feliz com o engajamento de todos vocês, então agradeço em nome da STEC e da Tecniplast. E a vocês, né, Ives, ao ICB, ao USP, pela abertura, na verdade, por esse espaço que vocês nos cederam, tanto para a palestra do Marco, quanto para essa nossa ação, junto aos participantes.
Então, vamos lá, só conferindo os comentários. Acho que teve muito comentário aqui que está carregando. Opa, agora foi. Confirmando certinho, só conferindo os comentários. Então, 195.
Bastante
Boa
coisa.
sorte, pessoal.
Vamos lá. Que rufem os tambores.
Olha, eu não sei quem... Olha, o pessoal se eu não me engano, é da Federal do Rio de Janeiro, de Macaé.
Que legal.
Vou dar um print aqui.
Vamos só
gerar
confirmar.
um certificado para ele. Priscila, você consegue conferir as regras aí, se ele atende?
Isso, estou conferindo já, Ives. Deixa eu só confirmar aqui no...
Enquanto isso, eu vou gerar o outro sorteio. Vou gerar o certificado para ele.
Leonardo Oliveira, né? Aprovadíssimo,
Isso.
Leonardo. Certinho, seguiu as nossas regras. Parabéns.
Tá validado. Ok.
Validado.
Vamos para o próximo. Ok, então tá validado. Vamos para o próximo sorteio.
Próximo.
Serão dois aquários seguindo o nosso sorteio.
Isso. Você não está participando não, né, Ives?
Olha, até comentei lá, mas tô brincando.
Vai que, né?
Tô precisando do aquário. Mais uma vez, agradecemos a presença de todos. Pessoal, esse foi uma ação nacional, então a gente teve a colaboração do pessoal da Stack. Vamos colocar os comentários, tudo certinho, fazer o outro sorteio.
Isso aí.
O Léo está presente na live? Eu não consigo, como eu estou fazendo sorteio, eu não consigo ver.
Ah, é verdade, né? Tem esse detalhe também.
Cadê a Natália? A Natália é a orientadora dele, lá de Macaé, se eu não me engano. O bom do Zebrafish no Brasil é que a gente acaba conhecendo todo mundo.
É verdade, conecta todo mundo, né? Ele é da
Sim,
UFRJ?
é Macaé. Vamos lá, fazer o segundo sorteio. Olha, a Ana Luchiari. Olha que beleza. Então, o segundo vai lá para o Rio Grande do Norte.
Aê, parabéns, Ana, que legal.
Confere aí, vê se tá tudo certinho, se ela seguiu as regras, e a gente pode
Certíssimo
continuar
também.
para.
Certíssimo também, Ana. Que bacana, que bacana.
Muito obrigado,
Muito bom,
pessoal. E
Ana,
agora
parceira
a
nossa
gente pode
também.
voltar pro inglês e continuar o nosso evento. Muito
Parabéns,
obrigado a
Leonardo.
todos.
Parabéns, Ana. Obrigada, Ives, pela oportunidade. Tchau, Marco. Thank you
Thank you.
for your partnership.
Obrigado. Thank you.
Thank you.
Thank you.
All right, guys. So now I would like you to introduce the next presenter for today's webinar. Okay. So Professor Leonardo Valdivia, he's an assistant professor at the Center of Integrative Biology at Universidad Mayor in Chile. His lab, the Developmental Genetics Lab, is focused on the study of fish eyes. So for this research, he has already been awarded with many, with diverse fellowships and awards, including the Best PhD Thesis Award by the Chilean Society of Cell Biology and the Early Career Stage Scientist Award at the Genes, Epigenetics, and Evolution in Eye Development and Disease Conference. Okay? So I would like you all to welcome Professor Leonardo. So Professor, feel free to start your presentation. And just to remind you guys that you can send your questions on our YouTube channel because we're going to answer them at the end of the presentation, okay? So thank you very much, Professor, and welcome.
Thank you, Claudia. I'm just going to share my screen now. Okay. Can you see that?
It's loading, yes. Yes, it's ready.
Okay, great. Give me a second. Okay. Well, thanks very much for the organizers for this invitation to present the things that we do in the lab. So today we are going to be talking about eye development. And the eyes are amongst the few noticeable organs that even a naive observer could spot in any vertebrate embryo. So it's not surprising that the eye has been a subject of developmental studies for over 100 years. So for you to just know, I mean, we are going to set the limits here. So these are two zebrafish embryos, and you can easily perhaps spot the eye here in this wild-type fish. And on the right, you have a mutant fish that display eye malformations. If you are able to spot this difference, I mean, you're ready to join the lab and study eye formation. So today, I'm just going to give you a quick overview of what we do in the lab, which special focus on novel gene trap and enhancer trap lines that we use to study eye formation in zebrafish, and this is part of a collaboration with Professor Koichi Kawakami, who is, you know, the father of the transgenesis in the zebrafish. So we're gonna divide this talk like in three main points that I'm gonna introduce in a few slides from now on. So before starting, like with this talk, I would like to highlight the key steps during eye formation in zebrafish. So I prepared this cartoon here in which we have some lateral views at the top and dorsal views of zebrafish embryos at the bottom. So there are three key steps that I would like to highlight. The first one is the evagination of the so-called optic vesicles. And for achieving that, we need to know that the eye-fated cells are established as a single domain within the anterior-most neural plate. So after a dramatic process, like this initial single domain is going to split into optic vesicles that we are showing here. And these optic vesicles will undergo further morphogenesis to acquire a three-dimensional shape, very similar to a mushroom head, right? Which is I'm showing here. And after that point, multipotent progenitors are going to start exiting the cell cycle to acquire postmitotic fate sequentially with some specific order for birth of these neurons. So the first neurons that are going to arise in the developing eyes are the retinal ganglion cells, which connect the retina with the processing centers in the brain. And at the end, we will get like glia and photoreceptors, and in the middle, we will generate a wide range of different kind of neurons. Okay, so just for you to know like how these three steps are just taking place, I'm just going to show you a time lapse taken from a paper by Jochen Rydberg lab in 2006, in which we will see this process of optic vesicle evagination. So you can see here that all the nuclei are labeled in red, and eye-fated cells are being shown in green here. So you can see that actually this single domain is split into optic vesicles. Later on, these optic vesicles will take like a three-dimensional shape, as I was telling you. And this is a second time lapse that I took from a really nice paper by Christine Kwan in 2011, in which you will see in green the a membrane form of GFP, and in magenta you will see the nuclei of the cells. So you can see that cells will start moving around the rim of the eye to feed with new cells the growing retina. And at the end of this process, we will ended up having something like I'm showing here. Okay, so you can see the developing eye here, and you can see the lens also just taking shape in this area. So after this process, differentiation will take place in the developing eyes. And so I just took this third time lapse from this paper by Bill Harris' lab, Alexandra Almeida in 2016, in which they use the SOFA approach, in which they show like that a combination of different transgenes in the same animal can be like a really nice way to document differentiation in this context. So you will see that there are some retinal ganglion cells in red. In green, we have some like different kind of neurons, and in the most external part, you will see the photoreceptors. So all these processes like are highly connected, and the things that I'm going to show you today, most of them are not published yet, but we'll touch like different steps of these movies that I'm showing here. So the question that we have in the lab is just very, very basic. So we are interested in understanding how does an eye form. And for that, we are using three complementary approaches. We are using forward genetics, in which we go from phenotype to gene. We will use also reverse genetic, going from gene to phenotype. And the third approach is using novel genetic tools, in which we have been isolating novel transgenic lines that are useful tools for understanding how the eyes are just taking shape. So the first one, I'm just going to state one just want to take one slide for just telling you like about this forward genetic approach. And this is just one sample of paper that we published when it was a postdoc in Steve Wilson's lab in London, at University College London, in which I participated in a forward genetic screen. So to make the long story short, we isolated a relatively large number of mutants affecting or showing defects during eye formation, essentially eye growth. Okay, so as I told you at the beginning, it's very simple for us to spot the difference because the size of the eye in the zebrafish in normal condition is pretty invariable. So any modification of this like shape of the size is like is very easy to spot. So we isolated one mutant that was displaying small eyes, and as I told you, to make the long story short, we found a mutation in GDF6A, which is a member of the BMP TGF beta family of proteins. And that gene, when it's mutated in human, this result in microphthalmia, which means small eyes, and people are born blind in most of the cases. So we found that actually this gene is antagonizing some signal that is very well known during development, which is retinoic acid, which is produced ventrally by the eye. And GDF6A is produced or is expressed in the dorsal retina. So these two signals are just antagonizing, and they provide some nice like homeostatic mechanisms to ensure we have like an eye with a normal size and with the correct number of neurons within this organ. So if we look at this animal that is around 3 days post-fertilization, and this is the eye, and we make a section here and we flip this eye in 90 degrees, we will be showing something like we see here. In red, you will see the nuclei, and in green, you will see the GFP expression driven by the regulatory sequences of the atonal 7 gene, which is a gene expressed specifically in the retinal ganglion cells, which are located in this layer of the retina. And actually, you can see here the axons just exiting the retina. And this is the wild-type condition. This is the mutant one in which we get like a small eye, but you can see that actually there is a overflow with neurons. There is an overproduction of neurons. Okay, so to analyze this process, like how this is just taking shape in vivo, what we did was to do time lapse, because I mean fish are just very good for that. So we can take our fish, put them under the microscope, and watch before our very eyes how eyes are just taking shape. In this case, this is a time lapse that took like 10 hours. I'm sorry, I put that in Spanish. But this is started at around 28 hours post-fertilization, just at the moment when the retinal ganglion cells, which are the first cells to be born in the retina, are starting to differentiate. So these animals were like at one cell stage were injected with a nuclear form of RFP. So we can see all the nuclei in red, and we will see the atonal 7 or AT5 GFP, which is the same one that we use here, as a marker for differentiation. So in this case, we can see that differentiation is just taking place normally in a clockwise manner. And this is the mutant condition that you will see that the eyes are just small and they overproduce like some neurons. I have to say that these two animals were observed or imaged in the same microscope at the same time. So these are just pretty comparable conditions. So the message in this paper was that we are like GDF6A is part of the homeostatic mechanisms that ensure we have the correct proportion of neurons in the eye. So if you don't have GDF6A, what is happening is retinoic acid is just pushing the cells towards differentiation without leaving time for them to proliferate. Okay, so we get like a small eye with an overproduction of neurons. And that's one message. That's an example of how we are analyzing these mutants coming from forward genetic screenings. The second one is about like the second approach, as I told you like a few slides ago, is about reverse genetics. We are using the well-known CRISPR-Cas machinery that, as you might see, like is very, it's a system that came to democratize the access to genetic manipulation, and actually by George Church has been told to be like a sort of genomic vandalism. Okay, so I'm just taking this image from this stencil by Banksy, in which we are just dropping these Molotov cocktails into defined places into the genome to modify local sequences, and essentially trying to eliminate the function of genes to reveal what's the function or what's that protein doing during normal development. And we have like a considerable experience with that. We have been doing or I participated in many papers in which we have been using that things, and even in our lab now, we are performing like medium to large scale screenings for identifying novel controllers of eye growth in zebrafish. Okay, so this is just one example of the mutants as we have been generating using CRISPR-Cas9. In this case, we generated a knockout in the MAP21L2 gene, which is a very important gene for eye development, but we don't know what's the molecular function yet. So what we know is when MAP21L2 is mutated in humans, people similar to GDF6A, they display microphthalmia, which means small eyes, and they are blind. The knockout in mice also display microphthalmia. We have generated this knockout allele in zebrafish, and they display small eyes, and it has been also shown by other groups, like including Elena Semina and Jeffrey Gross. Like different alleles are just showing comparable phenotypes, but also we have been just going a bit forward with that, and we have generated like a knockdown conditions in Drosophila for the MAP21 version of the fly, and the fly nicely shows small eyes also, which is something that is like the gene is just pretty well conserved during evolution. And this is the mutant condition. As you can see, they display small and flat eyes, and this is like one big part of the things that we are doing in the lab. So we are trying to understand how and why malformations arise in this context, and we are exploring that, trying to understand or using or studying cell biology in vivo in this context using light sheet microscopy, because recently, like a couple of years ago, we got funding, a really nice piece of funding for buying a size light sheet microscope, which is like a very unique way to look at this in the native context. Okay. So we are also just looking some cell-cell interactions or tissue interactions within the same eye, and we are also tracing the fates and also vandalizing the genome here to understand what is the transcriptional network driven by MAP21L2, because we have some evidence to think that MAP21L2 is controlling gene expression, but is not able to bind directly to DNA, but instead is just binding to proteins that bind DNA. Okay, so this is just for you to know the kind of things that we have been founding in this mutant, and this is the same transgenic line that I showed you before, the eternal 7 GFP, in which we are labeling the retinal ganglion cells and the projections to the processing centers in the brain, which is in this case is the optic tectum. And you can see that in the mutant condition there is like we get some differentiation. The eye is just a bit of malformed. It's has a shape that is similar to these really tasty tomatoes, which are just pretty deformed, right? But also we are seeing that just few axons are just just arriving to the processing centers in the brain. Okay, so this is something that has been done by Camila Baez Garrido in our lab, and we are just preparing a paper with this thing. And the third approach, which is the thing that I'm going to be focused now, is about this collaboration that we got with Koichi Kawakami. As you might have seen his nice talk, Koichi or Professor Kawakami has been doing a massive contribution to the zebrafish field, and not only the zebrafish, but mainly the fish model, in which he described this highly effective method for transgenesis based on the Toll-2 transposon. So he has been very, very generous. So I went to Japan a couple of years ago for a couple of weeks. So I had the opportunity to screen for quite a few transgenic lines. So I also brought back like a couple of things that I'm going to show you now. So even if we have like a nice collection of novel markers for the eye, I only just going to show you the data, the preliminary data that we have for 3 of these novel transgenic lines. Okay. So well, in basic terms, I mean, one of the nice contributions that Professor Kawakami has been doing to the zebrafish world is using the GAL4 UIS system for or trying to generate transgenic based on this GAL4 transactivator to be inserted randomly in different places into the genome. So this coding sequence for the GAL4 protein is also having upstream a minimal promoter, which is the place where the RNA polymerase is binding here. But that construct do not have any regulatory sequences. So if you drop that GAL4 cassette in a place in the genome where you don't have any gene in the surrounding, so that minimal promoter is not going to be active. Okay, so you will not get expression of GAL4. But if you drop this same cassette randomly, like close to a gene, the same regulatory sequences that are just controlling the expression of the endogenous gene are also going to work over GAL4 expression, which means that GAL4 is going to be produced in the same expression domains that the endogenous gene is being expressed. Okay, if you cross that line with a responsive line for GAL4, which is based on these UIS sequences controlling the expression of GFP, you cross these animals, what you will get is you will be able to report the expression of the endogenous gene based on this GAL4 UIS system. With that approach, that has been something like very, very successful because it's just very versatile. We can use GAL4 to control the expression of many different UIS-based transgenes. And this is one example of the things that we have been, or one of the lines that I isolated during my stay in Professor Kawakami's lab. The first line that I'm going to show you is tentatively named 268, which is a novel marker for developing Mueller glia in zebrafish eyes. And so this is a 24 hours post-fertilization embryo in which you will see the expression of the UIS GFP cassette, specifically in developing eyes. So in magenta, we're just showing neurons, so don't pay attention to that. But later on, at 33 hours post-fertilization, if we focus specifically in the eye, we get like expression all over the eye. But later on, 72 hours post-fertilization, where perhaps all the layers of the retina are just fully differentiated, like, and the eye is just starting to grow, we get this starry pattern of GFP that we know now that these are just Mueller glia. Okay, so we have like some evidence for that, which I'm not going to show here, but this is just for you to know, or just to look at this 3D reconstruction in which Mueller glia is just showing a very specific like shape, so we can easily spot them there. Okay, so we have this nice like transgenic line, which is labeling early progenitors in the developing eye, which is later on restricted to the Mueller glia. So as I told you, this is perhaps a novel marker for developing Mueller glia in the eye, because I'm just going to tell you what's the gene that is trapped in this condition. But also, I mean, this is additional data. Perhaps this is not the only place where this transgene is just driving expression, but also is like being expressed in some cells that seems to be microglia in the developing brain. So what we know now that this is like this transgenic line has an insertion of GAL4, like very, very, very close to this to a gene that has not been studied in zebrafish, which is called stonin-2. I'm not going to tell you details about it, but we have some molecular evidence to say that actually the GAL4 is just perhaps 100 bases away from the ATG of the gene. Okay, so it's upstream. And we have also evidence to say that this is not mutagenic, because it's like the homozygosis of the GAL4 insertion is not just showing any eye phenotype. Okay, but again, I mean, given that we don't have any functional evidence of this gene in the zebrafish, given that we had this experience with CRISPR-Cas9, we use these cocktail molotovs for dropping these CRISPR-Cas bombs into the genome, targeting specifically stonin-2, and we have found that actually loss of stonin-2 function leads to microphthalmia, as you can see here, small eyes in comparison to this control condition, and they affect Mueller glia development. And this is like Mueller glia, and you can see here that there is a dramatic reduction in GFP expression, and actually the shape of these cells are just very, very different. So this is the work, the PhD thesis that is just starting by Matias Benitez. So I hope in the future Matias will be able to tell you more details about this kind of thing. Okay. The second transgenic line, which is also an enhancer trap line, that remember, like when you have like insertion that is very close to a gene, you can just capture the genomic context for that, and though the regulatory sequences are going to act over GAL4. So in this case, we get a novel marker for retinal progenitors, but also tectal progenitors. And something that I didn't tell you is like the eye of the fish is just growing during the entire life. And the brain must be do the same thing, right? So to cope with this growth of the eye, the eye or the processing area of the brain must also be growing to adjust for those growth processes coming from the eye. So in this case, we have this tentatively named 370I GAL4 line. When it's crossed to UAS, we get some nice expression in retinal progenitor cells. So in this case, we are using an animal, or this is a confocal view or image, sorry, of 3 days post-fertilization zebrafish embryo, which is transgenic for this construction. And in blue, you will see the nuclei, and in green is the expression pattern of this transgene. And if we focus in this area, just around the lens is where the post-embryonic progenitors of the zebrafish eye are just living there. So they are just very close to the lens, and we can see that actually this expression is restricted to this area. And this is another view. In this case, this is a naked brain without skin and without eye. This is something that I learned from Kate Turner when I was in Steve Wilson's lab like a few years ago. And it's a really nice approach to like get some high resolution images of the zebrafish brain. And in this case, if we focus in the tectum progenitor area, we can get the mark of this transgene in this area. Actually, we are able to see some single cells. And the cool thing is, given that this is based on GAL4, actually we can, as I told you, we can just combine this transgene with some UAS RFP or K80 or different kind of cassettes in this context. And this is the work by Natalia Cruz and Bruno Maglione. So what we know now is like the insertion of GAL4 is very, very close also, but not interrupting the expression of this gene that is called ataxin-2, which is a gene that has been involved in ataxia in humans, but that again, it has not been studied like in zebrafish. So we have this like GAL4 like line in which like ataxin-2 is like in one defined area of the genome, and this GAL4 insertion is around 150 like bases upstream of the ATG, as I'm just showing here. And we have shown that like by sequencing and some molecular approaches. And then, as I told you, this is, seems not to be mutagenic, because when we get the homozygote GAL4 animals, they do not show any like defects in the during eye growth. But we have some evidence that I'm going to show you later that when you knock out the function of ataxin-2, actually you display, you show microphthalmia in this context. So, but this line is just very cool because we are interested in these kind of progenitors in the Zebrafish eye, and we are using this, like I put this cartoon here, to show that actually using light sheet imaging, we are able, or light sheet microscopy, sorry, we are able to report the behavior of these kind of progenitors. So in this case, I'm just showing here GFP expression in red, in which we have like this in vivo light sheet imaging movie that is just taking 10 hours. So what I want to show, this is not quantitative yet. I mean, we are just getting more numbers for this, but actually what we know, this is like indeed labeling progenitors, because these cells are just arising from this area. So they are just being added to the central retina. And also we have combined this transgenic light with some mutants that we have, in which we have an expansion of the progenitors in the ciliary marginal zone, which is this post-embryonic niche of cells that facilitate the growth of the eye. So we know that this is just behaving, this line or these GFP labeled cells are behaving as progenitors. They are just proliferating in this area, and they are contributed to the central retina. So this is a really cool like line for analyzing these kind of things in this context. And as I told you, I mean, if the hypothesis here was very simple, because if this, given that this gene is expressed in these progenitors, we hypothesized that if we knock out the function, we will get like sort of microphthalmia, and this is actually the case. So again, we are using this CRISPR-Cas approach, and in this case using crispants, in which we are just injecting CRISPRs to saturate with mutations a given locus, and in such a way that we will reveal the phenotype in the F0 that can predict what's going to be the stable loss of function condition in this context. And actually, this is something that we have quantified, and these animals display consistently small eyes. So this is the first functional evidence that this gene is like participating in eye formation in the zebrafish. And also we are collaborating also with Professor Janie Weeks at Harvard University, in which we have been like trying to do some human data combined with zebrafish in terms of the ataxin-2 function. This is ongoing work, so I hope in the future I'm gonna be able to tell you more details about it. And the third line that I'm gonna tell you about, like, is just very simple again. In this case, we have a mutagenic insertion of GAL4, which is not surrounding a gene, but in this case, it's just been inserted in the second exon of the RX3 gene, which is an important gene for eye formation. That gene works as a binary switch, in which if you don't have the expression of RX3, you don't have eyes, okay? So in this case, the homozygote animals combined with UHS-GFP, the homozygote insertion from GAL4 makes the embryos to lack eyes. And these are just heterozygote animals for this GAL4 insertion, again, combined with UHS. So we can report the expression of GAL4, but when the expression of RX3, but when you get the homozygote insertion of GAL4, you get an eyeless phenotype, but you can still report what happened with those cells that's supposed to be eye, but they are not eye anymore, okay? So we know now that those cells are being inserted, and they are being kept within the developing brain, and actually those brains are larger. And we are using this model to study, the neuronal basis of the genetic compensation phenomena, okay? So this is something that is being led by Rosana Reyes with who's the first postdoc that we have in the lab, but also with the contribution of Esteban Lira and Daniel Nahuelpan. This is like a front view of these same animals in which we are seeing the GFP expression here, but also in the hypothalamus. And here we get the expression, or sorry, the immunostaining against acetylated tubulin that is labeled in neuronal tracts, which is quite dramatically different in the mutant condition, in which we get this GFP area inserted within the brain. But also this line is just very good for like as a driver for IFA test cells. So we have combined that with UIS-CAID, and we are able to map the movements of cells that are just shaping the eye, in combination with one of the first mutant that I showed you a few slides ago, which is the MAP21L2 gene. So we are able, or we are hoping to be able to track the movement of these cells and how these like initial progenitors are being allocated within this post-embryonic niche of cells that I told you that actually is labeled by this ataxin to GFP transgene that I showed you a few slides ago. And this is the work by Camila Baez in our lab, who is a PhD student. So as a summary, well, I show you three complementary approaches to study the genetic basis of eye formation, but also the connection with growth and also the neuronal basis of genetic compensation, sorry, the sensory compensation. So in the first part, I just quickly show you some about forward genetics, in which mainly I showed some data about GDF6A, showing that there is an early depletion of progenitors in the retina in that mutant context. For reverse genetics, what I show you is like the function of MAP21L2 and what are the things that we are doing to understand how and why malformations arise in that mutant context. And the third approach, which is like the things that we are just strongly working now, is about the transgenic that we got in this collaboration with Professor Kawakami. So we have isolated two novel enhancer trap lines that are display GAL4 insertions close to the stonin2 and the ataxin2 genes in zebrafish, which are genes that do not have any functional data in this model. So these are two non-described genes in the zebrafish, which are just showing potential novel function. But also I show you some data about this gene trap line, which is mutagenic for this GAL4 insertion within the second exon of AREX3, which is facilitating the study of sensory compensation upon a genetic condition. So we are using all these transgenic lines combined with light sheet imaging to understand, how the eyes are just taking shape in this context. So before taking questions from you, I'd like to thank the people who did most of these things. So these are the current people that we have in our lab. At some point, we're eating here. At some point, we were like more people, but now, so we are just 6 of 7, taking myself into account. But also there are some other people that are just working in Joaquin Letelier's lab, who is a really good friend and collaborator at Universidad Mayor. But also I would like to thank Steve Wilson for all the support, and Gaia Gestri, who has been like a really, really nice person to speak about science also. Professor Koichi Kawakami, specifically Miguel Spigares, who was helping me when I went to Japan a couple of years ago. Dr. Elena Semina, who has been our collaborator in the MAP21L2 project, and the Zebraca crew. We call it Zebraca because we are working with zebrafish, but also Joaquin Letelier is also working with Medaka. So we are just sharing our fish facility, and we are just hosting the fish together. These are funding bodies, and of course, I would like to thank you for paying attention to this presentation. Thanks very, very much.
Amazing presentation, Professor Leonardo. Now, Professor is going to answer some questions, okay? The first question is from Flavio. Have you made the time lapse of the Mirglia reporter? He is wondering about how those early labeled progenitors eventually become glia, or maybe it's different populations at different times.
That's a really cool question. So I'm just gonna just let me go here. So that's a really nice question, Flavio. We need to bear in mind that this is what we are seeing here is GFP expression. So we are seeing protein, and actually this is some experiment that we were looking with Matthias just yesterday, because we were looking at the in situ hybridization of the GFP transcripts, and actually it seems to be the expression of GFP is restricted very early during development. So of course we want to look at how this is just taking shape, but we haven't looked at that using light sheet imaging. So I hope I'm going to be able to answer that question in the near future. Flavio, thanks very much.
All right, so let's see if we have any more questions. I don't think so, but everybody's complimenting the presentation. Okay, I'm sure everybody enjoyed it. It was a pleasure to have you with us. So thank you very much again, Professor Leonardo. Okay.
Thank you very much, Claudia.
So our next presentation is going to start at 11:30 a.m. Okay. So let's see. I think we can have a break now. Is it okay, production? Yes.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community. And we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us. Because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Saúde, direito de todos e dever do Estado. Direito à moradia, ao emprego, à educação, a um meio ambiente saudável, à água potável, ao lazer, à cultura. Direito à voz e à paz. Respeito à diversidade. Saúde para a Fundação Oswaldo Cruz abriga todas essas dimensões. É assim desde 1900. Uma visão integrada da saúde. Na pesquisa, ensino, assistência, informação, comunicação, memória, na promoção da saúde, na inovação, onde permanentemente se pensam respostas para as necessidades da sociedade, onde se faz ciência em defesa da vida. Você pode até não saber, mas carrega a Fiocruz dentro de você. Desenvolvemos e fabricamos vacinas, medicamentos, biofármacos, testes para diagnóstico. Formamos pessoas do nível médio à pós-graduação. Realizamos pesquisas para superar ameaças à saúde, para diminuir riscos ambientais, para prevenir doenças e agravos. Pesquisas que beneficiam crianças, adultos, idosos. Uma instituição pública e estratégica de Estado, integrante do Sistema Único de Saúde, com uma rica história de contribuições à sociedade. Presente de ponta a ponta no Brasil, onde cada trabalhador é um elo forte e ativo. Nela, ciência e saúde cumprem uma função social para o país e o mundo. Pés fincados na tradição, olhos voltados para o futuro. Somos patrimônio da ciência e da saúde, da humanidade, do povo brasileiro.
Situada no estado do Rio Grande do Norte, no Nordeste brasileiro, a Universidade Federal do Rio Grande do Norte está entre as maiores universidades da região. Fundada em 25 de junho de 1958, possui uma área construída de aproximadamente 300 mil metros quadrados, que compõe os 5 campi distribuídos por todo o estado. Tem hoje mais de 40 mil estudantes divididos em mais de 110 cursos de graduação, mais de 90 cursos de especialização, incluindo as residências, 80 cursos de mestrado e 40 de doutorado. Conta com mais de 2.300 professores, dos quais mais de 1.500 com doutorado. São mais de 1.900 projetos de pesquisas desenvolvidos pelos mais de 340 grupos de pesquisa cadastrados no CNPq, mais de 700 projetos de extensão e 4 programas permanentes, além de mais de 3.000 servidores técnicos administrativos. Como uma universidade de ampla atuação, as atividades de ensino, pesquisa e extensão se estendem a todas as áreas: ciências humanas, ciências sociais, ciências exatas e da terra, ciências biológicas, ciências da saúde, tecnologias e artes. Entre as áreas de destaque da UFRN, podemos citar o Instituto do Cérebro, que realiza pesquisas na área de neuroengenharia para o tratamento de doenças vasculares, epilepsia e outros distúrbios neurais. O Instituto Internacional de Física, que promove a troca de conhecimento científico com a comunidade internacional em áreas estratégicas. O Núcleo de Petróleo e Energias Renováveis, que por meio de parceria com a Petrobras, é considerado um dos maiores polos de pesquisa na área, formando mais de 1.000 profissionais em níveis de graduação e pós-graduação. O Instituto de Medicina Tropical, que tem por objetivo o estudo de doenças infecciosas com maior incidência em continentes de clima tropical, como América do Sul, África e Ásia. O Instituto Metrópole Digital, que atua na formação técnica e acadêmica por meio da inclusão social e digital de jovens, desde o ensino básico até a pós-graduação. A Escola de Música da UFRN, que tem por objetivo educar, produzir, disseminar conhecimento e fazer música como forma de contribuir para o desenvolvimento humano, promover a justiça social, a democracia e a cidadania. Nossa escola acolhe mais de 20 grupos e a Orquestra Sinfônica da UFRN, uma das únicas orquestras universitárias do país. O NUPLAN, Núcleo de Pesquisa de Medicamentos e Alimentos, um laboratório oficial destinado à produção de medicamentos para o Sistema Público de Saúde brasileiro. Entre suas atividades, o NUPLAN também oferece suporte acadêmico para a graduação e a pós-graduação, promovendo o ensino, a pesquisa, a extensão e a inovação tecnológica. O Ágora, Instituto de Línguas, Literaturas e Culturas Estrangeiras Modernas, que tem como principal objetivo oferecer cursos de língua estrangeira para estudantes, técnicos administrativos e professores da UFRN, bem como testes de proficiência para possibilitar a mobilidade internacional na universidade. O Ágora também recebe estudantes e pesquisadores das instituições parceiras da UFRN e de outros países, por meio do curso gratuito de português para estrangeiros. Na extensão, o programa de maior abrangência é o Trilhas Potiguares, que leva o conhecimento de professores e alunos aos municípios do interior do Rio Grande do Norte, em ações sociais desenvolvidas em parceria com as prefeituras municipais. Com mais de 20 anos de atuação, o sucesso desse programa já o levou também ao exterior, com a ida de nossas equipes a Moçambique, atuando com as equipes locais. Outra ação de relevo da instituição é a Semana de Ciência, Tecnologia e Cultura da Universidade Federal do Rio Grande do Norte, maior evento de atividades científicas, tecnológicas e culturais do Estado e um dos maiores palcos para a interface da UFRN com a sociedade. Em sua estrutura, a UFRN também dispõe de 3 hospitais universitários, 2 deles na capital e 1 no interior, 22 bibliotecas, 3 museus, canal de TV digital, rádio, agência de comunicação, editora, núcleo de arte e cultura, núcleo de educação infantil, residência e restaurante universitário. Em seu polo esportivo, disponibiliza campos de futebol e de treinamento oficiais, ginásios, piscinas olímpica e semiolímpicas. Assim, a UFRN busca a excelência e a inovação acadêmica por meio da formação de profissionais capacitados e ações educacionais comprometidas com a produção de conhecimento articulado com a integração social e comunitária, desenvolvendo soluções para os problemas regionais, nacionais e internacionais.
generate, and spread knowledge to form transforming agents. This is the goal of the biggest university of Rio Grande do Sul State Northern Region and one of the biggest from the country's southern region. With half a century of history, UPF maintains its roots in educational excellence, research and extension incentive, and the constant search for technological innovation. There are 9 campuses that are distributed around the region and 12 academic units, offering undergraduate, specialization, master's, doctorate, and postdoctorate courses. The infrastructure is a great differentiator. There are clinics, rooms for practical experimental teaching, laboratories, amphitheaters, auditoriums, and libraries with hundreds of thousands of titles available, both in physical and digital format. UPF stands out for the quality of teaching, academic, investigative in nature, the dialogue with the community, the performance in the job market of professionals who have graduated from this institution, and by the qualified teaching staff with teachers who promote the renewal of methodologies for the teaching and learning process. The research incentive in the university generates and disseminates knowledge. The extension reinforces the university's commitment to the community, with projects and programs that, through services provided, approximate academics to social reality and contribute to the sustainable development. Along with a university's social responsibility policy, the institution promotes citizenship, environmental awareness, social inclusion, culture, and development. Major events are held at the university. An example is the National Journey of Literature, one of the greatest literary manifestations in Latin America. The Radio UPF and UPF TV broadcasters with open signal and educational character provide the community with cultural programming focused on local development. Facilitating access to higher education, the university has several lines of credit, scholarships, and funding, benefiting more than 70% of students. One of them is the PAE, UPF's own financing program. Every semester, possibilities of exchange are offered to students through agreements with institutions in several countries. Acknowledgements strengthen the greatness of UPF. The university is among the best private institutions in Brazil. Its courses received grades 4 and 5 in the assessments made by the Ministry of Education. The institution has also received in recent years other important awards, such as Social Responsibility, granted by the State Legislative Assembly. The Planalto Medio UPF Scientific Park contributes directly to the development of the region. Connecting innovation, science, and technology, the UPF Parque, with its technology-based incubator, supports and encourages innovative projects and local entrepreneurship. Providing quality of life, UPF offers to the academic community services that favor inclusion and permanence in higher education and the construction of great stories. The university has a community center, a leisure environment to eat, interact, and rest. This is the University of Passo Fundo, an institution that prioritizes ethical and humanistic formation. UPF, knowledge is our nature.
When you walk into a pharmacy, over 90% of the products on the shelf have been tested on animals, such as rats, rabbits, and primates. These animal models have the complexity one needs, but they are costly, time-consuming, and ethically challenging. Also, 9 out of 10 products tested in animals fail when administered to humans. Even with biotechnological advances, the use of these animals in product research and development is still massive. Is there no other alternative to the use of these animals? And to what extent is maintaining this model worth it? Today, regulatory bodies pressure companies to change this scenario. In this perspective, large companies such as Natura, Boticário, and others are adopting an alternative methodology to replace the use of animals in tests. Thus, to offer innovative solutions, ZSafe was born. We are committed to using fish chambers for the toxicological evaluation of new products. It is an innovative, alternative, and predictive model. The fantastic thing is that, despite not being considered an animal model, this embryo shows cellular activity, guaranteeing the same reliability in animals and greater than tests in cells. Our tests reduce research time and costs in new product development by more than 50%. We have the perfect formula to provide greater profits for companies and reduce any possible ethical issues regarding animal use and welfare. So it's a win-win relationship. Our differential in the market is the competitive price, shorter service time, and delivery of reliable results. We have a multiple competent team that focuses on innovative solutions for the market and works to generate biomedical research safely and efficiently for the good of our customers, society, and animals. We are disease safe, and we are swimming towards a safer future.
My name is Mark Francis. I'm the founder of Aquatic. For the last 20 years, Aquatic has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community. And we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us. Because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps, and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Hello everyone again. I would like to introduce you now our next presenter, Professor Dr. Jennifer Hocking. She's an adjunct associate professor at the Division of Anatomy at the Department of Cell Biology from the Faculty of Medicine and Dentistry at the University of Alberta. Her lab studies the mechanisms underlying formation of an outer segment during development and its ongoing maintenance in the adult. This is in order to understand more about the cell biology of retinal dystrophies through manipulating zebrafish genetics. Currently, she mainly studies the role of actin cytoskeleton in the outer segment stabilization and renewal, and how neural activity influences cell morphology. She studies mainly the human disease cone rod dystrophy with a supernormal rod response by using the zebrafish model. So welcome, Professor. Hope you will enjoy the presentation.
Okay, thank you very much. Not sure. I'm trying to see my slides here now.
The screen sharing is not working so far.
Okay, that just shares that. That's not good. Okay.
Let's see. I think one of the organizers is going to talk to you.
I got it now.
Yep. All right.
Yeah. Okay. Can you see that?
Not
or
yet.
not?
No,
Okay.
It's loading. Yes.
There we go. Good.
Thank you very much.
Okay. And can you see my my pointer here too?
Not the pointer, only the screen.
Do you have a tip for how I can do that?
Let's see. I'm going to check with the guys. No tip.
Hi.
They're going to talk to you.
I guess the pointer would only work in the screen share, not in the upload.
Okay.
The screen share had worked actually when you try to screen share your screen. It was working here. We saw it. Maybe if you want to try it.
Okay, I'll try it one more time.
Yes, and then the video will work also.
Okay.
Yeah, we are seeing your screen.
Okay.
Your whole screen. So just put the...
Can you see it now?
Yes.
Okay, perfect. So hello, everyone. And thank you to the organizers for inviting me to talk today and for organizing this really impressive event. Before I even get started, I wanted to give credit to the student who did the vast majority of the work that I'm going to present. So rather than putting her name on every slide, I thought I'd give her a shout out right at the start, and that's Masha Sharkova. And not only did she collect the data, she also did all the digital art that I'm going to show you. She did get some help from an undergrad, Konstantin Muzaber, and I won't show you everything he did, but he did do the cloning that I'll mention later on. So in our lab recently, we've spent a lot of time talking about the apical surfaces of sensory neurons and epithelial cells. Okay, and it turns out this is a really interesting region of the cell
because it's not just a flat surface. There tends to be protrusions, different kinds of protrusions, and they look different from cell to cell. And they have different purposes. So they can have different sensory purposes, or they may be involved in increasing surface area for nutrient absorption in the intestine, for example, or they can release signals, release exosomes. So these protrusions can be involved in all sorts of different things, different functions, and they can look different, but they have all the same basis. So they're generally one of two types. Okay, they could be based on cilium. Okay, they can be a cilium or based on a sort of modified version of a cilium, which is what's shown in yellow in these diagrams. And so a cilium has it's a protrusion with an axoneme core, the axoneme being a ring of microtubules. And that ring is then anchored inside the cell at a basal body. The other main type of protrusion are finger-like projections known as microvilli. And these instead have a different cytoskeletal core. They have a bundle of actin, okay? And they tend to be smaller. So you can see on these different cells, this is a photoreceptor in the retina, a hair cell in the ear, a cerebrospinal fluid contacting neuron lining the central canal of the spinal cord, and a brush border cell, which could be in the intestine or in the kidney. And they have a combination of these, cilia and microvilli, and often both. And it's typically one is the major functioning part of this domain, but the other one's often still there, doing some sort of support role. Okay, and often we ignore whatever is secondary. Okay, so a really interesting example is if we compare and contrast the photoreceptor in the eye, the light detector, versus the auditory hair cell that detects sound. Okay, so the hair cell has a staircase-like arrangement of microvilli. Okay, they're actually called stereocilia, which is not a very good name because they're not cilia, they're microvilli, or expanded large microvilli. But they're the real mechanosensors. They're the real sound detectors. Okay? There is a cilium, though, known as the kinocilium, that is there developmentally. Sometimes it's maintained later on, and sometimes it's lost. So it's considered to have more of a support function. The opposite is true in the photoreceptor. For the photoreceptor, the sensory ending, the light detecting part of the cell, is based on a cilium. Okay, it doesn't look like a cilium anymore, but that's where it came from. Okay. On the other hand, there still are microvilli-like protrusions here. And so it's the opposite. Now the microvilli are thought to have the support function. Okay. So this kind of combination of different types of processes to perform the different functions of the apical part of the cell is interesting. And usually we just focus on one. We don't look at the combination in whole or how the two interact. And so in my lab, we focus on the retina. So I'm going to talk more about the retina and the photoreceptors. Okay. So here's a sagittal section through an eyeball. Okay, so light enters through the cornea and the lens and is focused onto the very back of the eye, where we have the thin layer that is our retina. Okay, the neural part of the eye. And the retina actually isn't a single layer, it's multiple layers of neurons, as well as a pigmented layer at the back. Okay, so light actually has to go through those first two layers to get to the photoreceptor layer here. Okay, the most outer layer of neurons. And the photoreceptors use their sensory endings to capture light. They capture photons and then turn it into a neural signal. Okay. So in the next slide, I want to take a closer look at the photoreceptors. Okay. So this is the general plan of how photoreceptors are arranged. They're very unique cells. Okay. And we have two main subtypes, cones and rods. And they're named cone or a rod based on the shape of their sensory ending. So the cone one tapers, it's more cone shaped. The rod one is just elongated like a rod. Okay. Cones are what we use for our high acuity color vision and central vision. Okay. Whereas rods, they're more on the periphery of the retina, and we use them in low light conditions. They're very sensitive, but they also saturate quickly. So both of them have this sensory ending, which we call the outer segment, that is a cilium-derived structure. So you can see the axoneme right here is forming the backbone, anchored to the basal body. But off of that is this huge expansion. And this expansion is a stack of membranous discs, stacked like a stack of pancakes. Okay. The rod one is a little different because they're individual pancakes encased in the plasma membrane. Whereas for the cones, they're all continuous. So they're more like lamellae, okay? Continuous and open to the exterior. But in both cases, the point is that we have a lot of membrane there, and that membrane is packed with components of the phototransduction cascade. Okay? Most importantly, with opsin molecules. And opsin is what interacts with the light, okay? Capturing that photons and triggering a neural response. Okay. So these outer segments are connected to the rest of the cell through a narrow bridge called the connecting cilium. Okay. And below that, we have the inner segment of the cell. And the inner segment is the powerhouse of the cell. These are very metabolically demanding cells. So there's a huge cluster of mitochondria. Okay. And the inner segment also makes all the protein and the membrane needed for the outer segment, which is a lot. It's a lot because we constantly turn over our outer segments. So we can't regenerate these cells. Fish can, humans can't. But in either case, we're constantly turning over these outer segments. And we do that by shedding the discs of the distal tip and synthesizing new discs at this proximal region. Okay. So other than that, there's a nucleus and a short axon and synaptic ending here. Now, the other player to bring in are those pigmented cells I mentioned. So just external to the photoreceptors, we have a layer of pigmented cells known as the retinal pigment epithelium. It's a single cell layer thick, but it's really important in maintaining the health of photoreceptors. Okay? And it does that in many different ways. So one is that they're pigmented cells, and that means that they absorb stray light. Okay? A second function is that they phagocytose and break down the shed outer segment discs, which otherwise would build up within the tissue. They also do many other functions, but those are two key ones. So what does it look like in zebrafish? Well, this is a nice diagram of the zebrafish photoreceptor layer. So not the whole retina, just the photoreceptor layer with an RPE cell added here. Okay, and this is modified from a Corbo Lab paper. And what you can see is here's a rod cell, which has actually a very thin region of the inner segment because the cell bodies are in this lowest layer, the photoreceptor layer, but their outer segments are pushed most distally. Okay, so there's this thin myoid region. Beyond the rods, there are 4 different types of cones. So in humans, we have red, green, and blue. In fish, there are red and green, which are actually fused double cones. There are blue cones, and there are UV cones. So fish have a wider spectrum that they can see than we can. Okay. And this is a section just showing bright field microscopy, that layer of the zebrafish retina. And glowing in orange here are labeled rod cells. You can see their nuclei. They're very thin myoid inner segments. And this is the ellipsoid part of the inner segment right before the outer segment starts. But the outer segments are pretty buried in the processes of these retinal pigment epithelial cells. So what I just told you was what you would usually hear if someone is giving you a description of photoreceptors. Okay, there's been a lot of focus on that outer segment, how it's formed, how it works. What people don't usually talk about are the other types of processes here. Okay, there are microvilli that surround the base of the outer segment. Okay, so they extend from the apical part of the inner segment and grow upwards, and they form like a cup around the base of the outer segment. And for that reason, they're known as calyceal processes. Calyceal means cup-like. Okay, and they're a type of microvilli, and you can see that here because they based around an actin core. Okay, that's what holds them, holds their structure. And that actin core doesn't end at the bottom of the processes. Instead, it extends as rootlets. Okay, so the actin extends as rootlets down into the inner segment and connects to this region known as the outer limiting membrane. Okay, so the outer limiting membrane is not actually a membrane, but in histology, it looks like a membrane, it looks like a line across the tissue section. And it's actually the site of cell adherence junctions between glial processes coming up from the middle layer of the retina and the photoreceptors. Okay, so there's a whole bunch of adherence junctions around this ring, and there's also an actin associated with that ring, and that ring of actin connects to the rootlets that become the cores of those calyceal processes. So if you look at different species, you can find these processes all over the place. So this is in primates, in a macaque monkey. They've got nice ones on this cone. This is in a pig, quite long ones. And this is in frog. In frog, they're associated with grooves called the incisors. And they're also there in humans. Okay, this has been demonstrated. What's interesting is they're missing in not all rodents, but it seems to be that they're missing in nocturnal rodents. And that's significant because they're missing in our rodent animal models, meaning mouse and rat, which is probably why these processes have been largely ignored in the literature. Okay, because they're just not there in the species we look at, or many people look at. So the question is, why are they there? What are they doing? Right? They're not the sensory ending. The outer segment is the sensory ending. It's a light detecting part of the cell. So why do we have these processes, especially if in some species they're not actually needed? Do they have a function, or are they vestigial? So to get, and is there any evidence that they are important? And the best clue we have is that they've been associated with Usher syndrome. So Usher syndrome is the most common form of combined hearing and vision loss. And the most severe form, known as Usher type 1, is caused by a number of different mutations in a number of different genes. And the protein from those genes it's well known what they do in the ear. Okay, they form complexes in the stereocilia that are important for the mechanotransduction process. Excuse me. Now, the thing is, when they make mouse models, they have hearing loss, but they don't have a vision problem. There's no retinal phenotype. And so the theory is that maybe it's because those rodents don't have these calocial processes, okay? Because the problem is in the microvilli of the ear, so could the problem be in the microvilli of the eye, okay? And in fact, the Petit lab has shown that these different proteins localize to the processes of different species, but here they're showing in macaque. Okay, so these little punctate are cross sections through processes around an outer segment. Okay, so there's some association there, but we really still don't know what these processes are doing. And we therefore turn to zebrafish to see if we can use zebrafish as a model to study them. And luckily, they're pretty easy to label because they're actin-based. And so here you can see that we've labeled actin with phalloidin, and you've got beautiful filamentous structures. It's not all the processes, but we can clearly see the processes here. So these little fingers right here, these are CPs of UV cones. Up here, these would be the CPs of double cones. And right here, you can see one around a rod outer segment. Okay, so we can label them. But before we start playing with them, we need a basic characterization. We need to kind of know the basic plan of what these are like, so we know what the difference between normal and abnormal is. So MASH has been doing a very detailed characterization of what these processes look like. Okay, and this is just one of her images, just to show you the kind of view she can get and the arrangement of actin in these cells. So what you're seeing, these rings down here, that's the outer limiting membrane. That's where the junctions are between the photoreceptors, and the labeled photoreceptors here were UV cones, and Mueller glia. Okay, and extending up from there, you can see fibers that are the rootlets. And then right here, this is junction between inner and outer segment. And you can see little fibers coming up from that junction. Those are the processes, the CPs we're looking at. Okay. So the first question was, how long were they? Okay. And so at 1 month, when these cells are pretty mature, she went through and measured the length of the filaments, okay, from the inner segment alongside the outer segment. She can measure them for rods, for double cones, and for UV cones. And they came out to be about 3 to 6 microns in length, which is pretty consistent with microvilli on other cells. What was particularly interesting here, though, was we then compared the length of the process to the length of the outer segment itself. And that's where we saw significant variation. So for rods and double cones, the CPs extend about 30% of the way up the length of the outer segment. So really around the base of the outer segment is where they are. In contrast, for UV cones, they extend almost the entire length of that outer segment. So they cover about 80% of it. Okay, which is interesting because does that suggest that they have slightly different functions for these two different types of cells? Okay. So the second part of the question, just looking at the length, is it static? Is the length always the same? And the reason we ask this question is because these cells, these photoreceptors, are quite dynamic. So in teleos, fish, frogs, and birds, there's something called retinomotor movements, where you get active movement of photoreceptors in response to dark and light. Okay, so this would be a dark adapted retina, and this is a light adapted retina, so DA and LA. In the dark, what you want to do is maximize the exposure of rods to photons. So they contract the rod inner segments and bring the outer segments closer to the light. You don't need the cones, so the cone inner segments elongate and push the outer segments farther away. Okay. The other change is the pigment granules. So you try to get the pigment granules out of the way of the light, and so they retract into the cell body for the retinal pigment epithelium. And then the opposite happens in the light. In the light, cones, the rods are not very useful, so they get out of the way. The cones are brought closer to the light, but there's also pigment granules in those apical processes of the RPE to help absorb stray photons. Okay, so there was one paper from the Burnside lab that showed that it's not just the cells that change dimensions, but the processes also do. So, and this is a dark adapted cell. This is I think from a sunfish. You can see very long processes. And in a light adapted cell, they're quite short. So this is interesting, but this is in vitro. These were isolated cells. Does this hold up in vivo? So Masha did very careful analysis of light and dark adapted fish. And the first question was, can she see the retinomotor movements themselves? So not even looking at the CPs yet, just looking at the cells. And indeed she could. And so if you actually look at the bottom here first, so this is dark adapted and this is light adapted. And it's really obvious between these two images where you can see that the cells have different lengths of inner segments, which positions their outer segments differently. And so she quantified this, and you can see a big change in the length of the rods. And as expected, those rod inner segments get longer in the light, whereas the inner segments for the cones get shorter. Okay, for the cones, it's less obvious, but it's a significant change. Okay, so she confirmed that the cells really were undergoing these retinomotor movements as expected. What about the CPs? Did they change in length? And in fact, she didn't see any change, not for the cones and not for the rods. The length of the CPs stayed pretty static, even though this inner segment region was expanding and contracting. It is possible, because we're looking at whole tissue, that we're missing a subtle change. But regardless, we're clearly not seeing that there's no dramatic change in length from dark to light. Now, one thing I wanted to point out is that in some cases, measuring these processes can be difficult, because if you get pigment granules from the RPE wrapped around the cells, then it obscures your point of view, okay? And it obscures the staining of actin or cell markers. And so what Masha did is she used crystal fish, which are triple knockouts and have no pigment, okay, including no retinal pigment epithelium. So these eyes should be black. Instead, you can see that they're transparent. And she had to do that to measure light-adapted rods, because light-adapted rods are buried in the RPE. Now, the concern was that these are not the healthiest fish. And so are we getting a real result here? So here you can see the beautiful staining she could get in these fish. So the first thing is the processes are clearly there. Okay? It's not like they're looking completely disorganized. They're looking healthy. But the other thing she did was she compared the processes between wild type fish and the crystal fish for dark adapted rods. So in this case, she didn't need the crystal fish to measure the length of the CPs. And she found that they were comparable. So this was arguing that the processes are pretty normal in the crystal fish. Okay, so arguing that this is real data that we were seeing, showing no change between dark and light. Okay, so the next question was, okay, we know how long these are, how many are there? And for this, she took sections in a perpendicular plane, so now cutting sagittal sections through the retina, trying to go right across that photoreceptor layer. And you can see these little punctae that are the processes. Okay, and so she was counting how many there were, and here you can see a blue cone. These DC is double cones, so these are the red and green cones that are fused together. And so she saw about 20 processes surrounding double cones and about 13 surrounding blue cones. She didn't get measurements for rods or UV cones because they tended to be thinner. Okay, so staining with actin, with phalloidin for actin, was a bit weaker, and it was hard to see individual punctae. So the alternative for that is to do TEM, and here's just a nice example where you can clearly see those processes sitting right beside an outer segment. So here's an outer segment. It's cut kind of an oblique angle, not straight across. But this is outer segment with layers of membranous discs in it. And this is mitochondria in the inner segment. So that's a work in progress at the moment. Now, zebrafish are well known for having a very organized photoreceptor layer. Okay, they have this mosaic pattern, so you don't have random placement of the photoreceptors within the retina. They're very orderly. Okay, so you have rows of alternating UV and blue cones, as well as rows of alternating flipped red and green cones, and rods in between. Okay, so it's very orderly, and you can see that clearly in her sagittal sections. Okay, so this particular image is taken at about this level. Okay, and what you capture here are, you capture the very thin myoid inner segment regions of the rods, and you capture the outer segments of the UV cones. Okay, so in this image, labeled in orange, those are rod inner segments. And then within the circles of labeled actin here, that's where you would have a UV cone outer segment. Okay. Now, if we blow that up, what we can see is that we're looking at a ring of CPs, calcial processes here. Okay, so you can see dots there. You can see places where it's harder to see individual dots, which is why we haven't counted these ones, but there's clearly some dots. So these are the processes surrounding the outer segment. And there's the rod inner segments. Now, what caught our interest was that didn't account for everything here, right? Because there's these larger blobs of actin that are clearly not the CPs. It's clearly a larger bundle of actin, and it's got to be something else. There's something else that is surrounding those UV cone outer segments. And so what could it be? And our first guess was that it would have to do with the RPE, those pigment cells, because they extend these apical processes. And some people call them microvilli, and in part they're microvilli, but they're more complicated than that. Sometimes they're more like lamellae wrapped around outer segments. What's interesting here is that the UV cones are a long way from the RPE. They're in this lower level of the photoreceptor layer. And so how far do those processes actually extend? Okay, and we do think they extend this far because we can see pigment granules in TEM there. But are they what are forming these actin bundles? Okay, are there actin-filled RP processes there? And so to test that, Masha did staining for phospho which in this case is labeling active ezrin. And we know ezrin is a marker for the apical processes of RPE. Okay, so in green is actin, and then the pink is the phospho-ERM. Here you can see the phospho-ERM on its own, and you can see how it's labeling thick filaments there. And it overlaps with those thick filaments of actin. So this made sense to us. So this must be RPE processes, which is neat because it's such a regular arrangement. However, when we looked at this more closely, when Masha looked at it more closely, what she noticed was, yes, they co-localize. Okay, so those thick actin bundles do co-localize with the phospho-ERM, but not entirely. So the red here is the co-localization, and then the yellow are the thick actin bundles that did not co-localize. So it's like the phospho-ERM just ended. The RP processes reach their limit. They don't go all the way down to that outer limiting membrane. So then what's the actin down here? And we were quite stumped for a while, but we recently came across or revisited this paper from the Way Lab, Pennsylvania, looking at the Mueller glia. So the Mueller glia sit in the middle layer of the retina, but they extend processes both directions, down and up. And as mentioned, those apical processes of the glia form junctions with photoreceptors at the outer limiting membrane. So right along this line. And it's well known that they have little microvilli that protrude upward from that. And that's what is labeled as short processes here. What's less understood is that there are long processes too. There's fewer of them, but there's clearly these longer, thicker processes protruding upwards as well into that photoreceptor layer. And in sagittal section, they look a lot like those thicker actin bundles that surround the UV cones. So what we think is happening is that we've got RP sending processes down, Mueller glia sending processes up, and the two meet. So they meet in such a way that they form almost what looks like a continuous bundle of actin. Okay, but it's not. It's actin within the Mueller glia, actin within the RP, almost perfectly aligned. And together, they form a cage around the photoreceptor, around the UV outer segment. Okay. And that's on top of the inner cage here made by the CPs. So it's almost like there's two layers of support for that outer segment. So the next question was, well, how dynamic are these? You know, are we constantly breaking down and remaking the CPs, or are they very static structures? And it'd be nice to do live imaging to see if which of those is true. That's pretty hard in these mature retinas. So the way that we're addressing it at the moment is asking, how dynamic is the actin? So how dynamic is the actin within the core of the CPs? Okay, how fast does it turn over? And there's two basic models for this. So the classic one is what was worked out in enterocytes in the brush border, where you can see the actin treadmills, meaning that you add new actin monomers to the filaments at the tips, the plus ends of the actin, which is in the tips of the microvilli, and remove actin monomers from the filaments at the minus ends inside the cell. Okay, and so you constantly turn over the actin filaments while at the same time maintaining the microvillus. Okay, so this is called treadmilling. And that was thought to happen in hair cells, but it turns out it's not the case. So in hair cells in the ear, they actually have really stable shafts. Okay, so the actin that forms the core, the shaft for the stereocilia, it lasts there for weeks and weeks. Okay, pretty much as long as it's been looked. It's only at the tip where the actin turns over. Okay, so this is the tip turnover model. So what is happening in photoreceptor CPs? Well, to ask this question, we're following one of the experiments that was done in stereocilia, and we're making an inducible tagged actin. So we use the TOL2 kit to clone actin, zebrafish actin, beta actin, behind the heat shock promoter, and we've added a MIC tag. Okay, and so this has been injected into single cell embryos together with TOL2 transposase, and we now have mosaic embryos and larvae and adults that we're analyzing to see how quickly the actin turns over. Okay, so when we do this in larvae, so these are fish that were injected at the 1 cell stage and then heat shocked at 5 days to induce the new actin. And then we gave them 24 hours as the opportunity to incorporate the labeled actin. So this is showing the photoreceptor layer where the new actin is labeled, and here it's showing phalloidin labeling. So all the existing actin, or all of the actin. And you can see 3 cells expressing the induced actin. And you can see where the actin has incorporated. Okay, so we can see it's incorporated at the synapse, or at the synaptic ending, where we expect it to. Interestingly, it's also incorporating in the processes. So at this point, the actin in the CPs is dynamic, and it's incorporating largely into the rootlets as well. Where it's not being incorporated is at the outer limiting membrane. So that outer limiting membrane is very stable at this point. Maybe not surprising, because that outer limiting membrane is established even before the cells differentiate. Those contacts are there. So this shows us that our construct works, that we can see incorporation where it's happening and where it's not happening. And it shows us dynamics early on. So in these 6-day-old larvae, this is about 3 days after the cells have been born. So they've had outer segments for a while. CPs are not new. And there's still dynamics happening there, but they're not adults. And so what we want to find out is what happens in the adults. And that is our something we're working on. It's just harder to heat shock adults. But we'd really like to see at some point, do these processes become quite stable, or do they continue to undergo renewal? Okay. And so that was later, but what about right at the beginning? And how early are these processes actually there? And particularly, are they there as the outer segment is forming? And so these are beautiful pictures from an old paper showing the chick retina. And so these are photoreceptor cells. It's the surface of the inner segment before the outer segment forms. And you can see all of these microvilli-like processes. So suggesting the CPs are already there. Okay. And here's a day later where now there's a cilium that's emerged and will become the outer segment. And there are the processes ready to support it. Okay. So in the fish, we've seen a similar thing. So these are labeled UV cones. And if you look at 68 hours, some cells are just beginning to form an outer segment, a tiny, tiny little one. And what we see with actin is that actin forms like a cap over the top of the inner segment. But even then, we can already see little protrusions. So there's some sort of little microvilli here. 72 hours, now we can see some clearly forming outer segments. And there's very obvious processes that are present. So CPs are there as the outer segments are forming. And we could see that also in TEM, where here's a photoreceptor, here's the inner segment. This is a emerging outer segment, which sometimes they just look bubbly as they're starting to make discs. And there's a process right beside it. Here, this is a little farther along with the initial stacks, initial discs of the outer segment, and there's a process right beside it. Now, we recently set up a collaboration with the Zolesi lab because they look at differentiating photoreceptors. So earlier on, and what they saw were very dynamic apical processes, microvilli, more like filopodia almost because they're so dynamic, but actin-based protrusions from the apical side of the cell before they even are differentiated as photoreceptors. So what we're trying to do now is fill the gap here, fill the gap between 2 days and 3 days. Because what we don't know is, do these protrusions turn into the CPs, or are they retracted and do the CPs emerge sort of together with the outer segment? Okay, lastly, the other thing to address here are what else is important. So I've talked a lot about actin, but it's not actin alone here that's important. There are all sorts of other protein components that support microvilli. And you can see there's protein components that have been identified for stereocilia, for the brush border, for cerebrospinal fluid contacting neurons, and for CPs. And some of these will cross-link actin, some will act like capping factors for the actin, and some of them form intermicrovillar links or links between cilium and microvilli. And so what are the players that are going to support them? And while some have been localized, we don't know much about what they could be doing in the CPs. And so the main one that's been looked at is protocadherin 15. So protocadherin 15 is thought to form part of a linker connecting the CP to the outer segment. And in frog morphants or fish mutants, you see disorganized or dysmorphic outer segments. So the outer segments can be bent, or you can see overgrowth of new discs. So instead of sort of reaching a limit for the width and then starting to add a new discs just keep growing, okay? Which supports the hypothesis that what these CPs do is form kind of a ring around to constrain growth and support the outer segment. But microvilli can do all sorts of things. So we don't know if that's the full role for the CPs. And one thing we want to do is we want to manipulate a lot of these components to see if we can change processes and in turn what that will do to the outer segments. So I'll stop there. What I've talked about today is that all zebrafish photoreceptors have these microvilli-like processes, about 3 to 6 microns in length, but how much of the outer segment they cover is variable. Okay, the UV cone showed has got a cage of these CPs around it, and then another outer cage that seems to be composed of Mueller glia and RPE processes together. And you can even add in the inner segments of the rods there as well. And it could be that the glia come into play because these cells are farthest from the RPE. So the other cells may just use RPE. We've shown that CP actin is dynamic in the larvae. We have yet to look in the adults. And we know that CPs are there early. So they're definitely there as the outer segments are initiating. And we're working on that transition as to when they actually first emerge. So I'll stop there, and I'd like to thank primarily Masha, who did the vast majority of this work, which is really detailed, but she's come up with some beautiful, beautiful images. Constantine for the work on the heat shock construct, and other members of the lab, and other support people at the U of A, and all of our funding sources. So I'll stop there, and thank you very much.
Thank you, Professor Dr. Jennifer Hocking, for your amazing presentation. I'm sure everybody enjoyed it too. I would like to ask the public if they have any questions. So just a minute. It is here. So Flavio is asking if UV cones seem to be more supported by actin-based processes than the others. Any guess on a reason for this need? They are closer to the OLM and shorter, maybe that?
We don't really know. And so it could very well be that because they're farther from the RPE, they're not as enwrapped by the RPE, but that's something we're kind of looking at. The interaction between the RPE and the photoreceptors, I think, is actually quite complicated and is very much oversimplified when you look at descriptions, because people focus on, oh, it's just a brush of microvilli. It's not really. In fact, they're not really microvilli at all, except that bit around the UV cone outer segments, because otherwise you don't have the finger-like projections that have a core of actin. They're much more film sort of networks of actin inside the RPE. So we have no idea why this is. That the fact that they're just in a different layer, because they're very much in a different layer than the other photoreceptors. And so maybe that's why, but it's a guess.
All right, thank you very much. So thank you again, Professor Jennifer. It was a pleasure to have you with us. And for the public, I just want to let you know that it was our last presentation for today's morning. Okay, we are going to be back at 2 p.m. Okay, live on the same channel, and hope to see you back here. Okay. Thank you very much.